So this is a video about bacterial cell wall synthesis. So the bacterial cell wall synthesis begins with NAG. So it begins with NAG, to which we are going to add or is added UTP. This results in the formation of UDP NAG. Now, for the sake of simplicity, we're going to represent this with this figure. Now, UDP NAG will then be converted to UDP NAM. What is NAG? N-acetyl glucosamine. What is NAM? N-acetyl muramic acid. So we convert the NAG to N-acetyl muramic acid in the presence of an enzyme known as enol pyruvate transferase, also known as MUR-A enzyme. So this converts this to this, which is N-acetyl muramic acid. Then the amino acids, which are part of the peptide chain, will be added to NAM. 3 of them will be added, which is L-alanine, D-glutamic acid, L-lysine. But the one which is missing is the alanine. How do we get the alanine? Well, we're gonna get the alanine from L-alanine. L-alanine will be converted to D-alanine as a part of a racemization reaction in the presence of an enzyme called as racemase. This converts L-alanine to D-alanine. Then 2D-alanine will form a dimer in the presence of an enzyme called as D-alanine ligase. So you get 2D-alanine, and then these 2D-alanine will then be added to the peptide chain to become a part of the peptide chain. Then what happens? Then this process, which started in the cytoplasm, now this complex form will move towards the plasma membrane. And here we are. Well, on the plasma membrane, there is an enzyme called as bacterial. The bacterial is a type of pyrophosphate, which means it has a lot of phosphate. Now, one of its phosphate will be used for binding to or as a target by our initiator complex. And this is the same complex that we had before. Okay, 5 amino acid part of the peptide chain. This is the NAM and this is UDP. Next, we are going to add this complex to the bacterial, or the phosphate of the bacterial. But before that, we actually remove the UMP. If we remove the UMP, we're left with a phosphate here. We are left with one phosphate here, not two as before. We're left with one phosphate. And that phosphate will join with the phosphate of the bacterial, and now we have our complex sitting on the bacterial. Whilst on the bacterial, on the inner side of the plasma membrane, right? So let's say this is the inner side of the plasma membrane. The complex will be modified. Well, not be modified, will be added to. It's going to be added to. What are we going to add to it? 2 things. First would be a NAG unit, a NAG sugar, the same thing that we had before. N-acetyl glucosamine will be added to the N-acetyl muramic acid through a glycosidic bond. And the second thing which will be added is the pentaglycine. The pentaglycine, which is responsible for the formation of cross-linking between different peptidoglycan chains, will be added to the peptide chain. Then the bacterial will transfer this from the inner side of the plasma membrane to the outer side of the plasma membrane. Once it's on the outer side of the plasma membrane, now we're going to have a step called as polymerization. Well, polymerization. What is polymerization? Essentially, here we have this complex, okay? Now we're just gonna extend it. So we had 2 particles or 2 sugars. Now we're gonna make it 5, 6, 7, 8, whatever. We're gonna extend on it. We're gonna form a new glycosidic bond. And also, we're gonna have the peptide chain sitting on the NAM. So this is just polymerization. Multiple units will be joined together. This process is catalyzed by an enzyme known as glycosylase. I hope I spelled that right. But anyways, so we have this complex sitting here. The next step is a step which occurs in gram positive but not in gram negative. What is this step? It's called as cross-linking. Cross-linking is done by another enzyme known as transpeptidase. Another name for this, another name for this is penicillin binding protein. Okay, so transpeptidase catalyzes the cross-linking between different chains of peptidoglycan with the help of pentaglycine. So the pentaglycine is what forms the cross-link between the peptide chains, and this is the entire step, or a brief version, you could say, of cell wall synthesis.